Good morning, students. In the first part of proteins, we have studied about the structure of proteins, about the various characteristics of protein, denaturation of protein, types of protein, etc. So, here we will study the structure of protein. As you all know, proteins are complex biological macromolecules which are composed of alpha amino acids. And these alpha amino acids are linked together by peptide bonds to form one or more polypeptide chain. Now, this simple structure will not explain the multitude of biological functions which a protein can undertake. So, this is not the exact structure of a protein. There are many hierarchical arrangements of amino acids which are essential for biological function. And these three-dimensional arrangements of these polypeptide chain determines the structure and function of proteins in living systems. So, the structure of protein can be categorized into four levels. One is the primary structure, which consists of linear sequence of amino acids. Second is the secondary structure, where these primary structure are held together by hydrogen bonds. These secondary structure aggregate to form tertiary structure, and the tertiary structure assemble to form the quaternary structure. And each of these structures, that is primary, secondary, tertiary, quaternary structures, determine the shape of a protein, whether it is fibrous or globular, and it is crucial for enzymes, antibodies, and the structural function of protein. Now, let's go see the structure one by one. So, the first is primary structure. Primary structure is the simplest structure of a protein, and here it consists of an amino acid sequence. That means the amino acids will be packed in a unique, precise order, and this will be linked by peptide bonds, and this order determines the identity of a protein and the function of protein. And the second point you have to take care of is that we already know these amino acids are held in a precise order by means of a peptide bonds. And these peptide bonds are covalent bonds which are formed by the condensation reaction between carboxyl group of one amino acid and the amino group of the other. What about the direction? Now we know this consists of long chains of amino acids linked together by peptide bond. But what about the direction? Directionality is always the N-terminal amino acid will be on the left and the C-terminus will be on the right. And the specific sequence, like if you have glycine, proline, threonine, again glycine, this type of specific sequence, it is determined by the DNA of the organism. And the determinant of shape, the linear structure, and this sequence as well as the linear structure determines how the protein will fold into the further hierarchical structures like secondary and tertiary structure. So, you have to take care of these points: amino acid sequence, peptide bonds, directionality, the genetic control, and the determination of the shape. Now, the importance of primary structure. primary structure gives a structural integrity, stability to the structure of the protein. But it can be broken by hydrolysis. Now, the second point is mutation effect. If there is a small change in the sequence, like change, if one amino acid is replaced by another, or if the order is not correct due to genetic mutation, then that can lead to misfolding. The folding won't be correct, and this may lead to the loss of the function of that protein and disease. For example, substitution of one amino acid in the hemoglobin molecule causes the disease sickle cell anemia. Moving on to the secondary structure, we know that primary structure is a sequence of amino acid. Now, more such primary structure collectively forms secondary structure. So, secondary structure is highly ordered, local, repeating three-dimensional folds of amino acid sequence or polypeptide chain, excluding side chain. You don't have to take side chain in secondary structure. So, primarily this secondary structure is held together by hydrogen bonds. Hydrogen bonds between the amino nitrogen and the carbonyl oxygen. And the type of hydrogen bond between the polypeptide chain determines whether the secondary structure arrange itself into helices or coil, or pleated sheet like foldings of paper, or turns and loops. Let us see in detail the main components of the secondary structure. We already said it can be arranged into three different ways: helices, pleated sheet, and turns or loops. Helices means this one, the right-handed helical coil. The amino acid sequence can arrange in this manner in the form of a right-handed helical coil. And this is held together by hydrogen bonds between every fourth. So, if there is a hydrogen bond start from this amino acid, then it forms the hydrogen bond with the carbonyl group of the fourth amino acid. So, hydrogen bond form between every fourth amino acid. So, it will naturally form a right-handed helix. Or two or more polypeptide chain can be arranged into a folded paper manner or a pleated sheet manner, either in the parallel way. The two can align in the same direction, that is the parallel arrangement, or in the opposite direction, that is the anti-parallel arrangement. Again, here it is stabilized, as you can see, by means of hydrogen bonds. Now, if segments allow the peptide chain to reverse the direction, if this peptide chain can reverse the direction, then it forms turns or loops. So, these are the main components of secondary structure. Now, the stabilizing force, is the hydrogen bonds, and the significance of secondary structure is that it forms the most important or the intermediate step in protein folding, or provides structural strength to fibrous proteins like keratin. Now, this collection of secondary structure forms tertiary structure. So, tertiary structure is a complex three-dimensional shape formed by the folding of polypeptide chain in secondary structure. That means helices or sheets can be folded into a three-dimensional structure in this manner to form tertiary structure. And the tertiary structure is stabilized by means of different types of interaction. And this interaction, it could be hydrophobic interaction, disulfide bonds, ionic bonds, or hydrogen bonds, and that defines its biological functionality. So, hydrophobic interaction occurs in the non-polar side chain inside the protein, away from the water. Disulfide bonds, that is in the case of cysteine amino acid, there will be strong SS bond, strong covalent bond between the sulfur atoms in cysteine residues. Hydrogen bonds, we know. Ionic bonds or electrostatic interaction between charged sites, we know all amino acid sequential chains contains two terminals, N-terminal and C-terminal. And the ionic interaction between the charged site forms the ionic bond. Now, tertiary structure is very important in terms of these points. One is the structure and function. It enables the functionality of proteins. Misfolding can lead to inactive or pathological proteins. Protein domains are created by tertiary structure, and it forms the building block of protein structure. The tertiary structure can be determined by X-ray crystallography, NMR, and cryo-electron microscopy. And the structure of our tertiary structure is stabilized by chaperone proteins, and these chaperone proteins are proteins which assist in guiding the protein to its most stable conformation, what we call native conformation. Now we come to the quaternary structure, which is the highest level of protein organization, and it simply is a collection of three-dimensional tertiary structure. It contains multiple folded polypeptide chain. They assemble, interact, and arrange themselves into a single functional multi-subunit complex known as quaternary structure. Now, subunit assembly, if you consider, they form dimer, trimer, or larger oligomer. In the case of functional role, it allows for complex functions like allosteric regulation, cooperativity, and formation of active sites at subunit interfaces. It is stabilized by weak non-covalent interactions which hold the unit together. And if for familiar example, hemoglobin, it contains 4 subunits, DNA polymerase contains 10 subunits, and keratin are some of the examples of quaternary structure. To sum up, this is the primary structure, where it's a linear sequential arrangement of amino acid. This combined to form a secondary structure. Here alpha helix is formed. Also pleated sheets, turns and loops are also possible. And these alpha helix join to form the tertiary structure. There is some kind of foldings involved. And these tertiary structure agglomerate or combine to form the quaternary structure. This is the structure of complex protein, and this is the X-ray crystallographic diagram of a quaternary structure of a protein. This is how the protein will look alike in real life. So I hope you understood the structure of protein. Structure of protein usually appears as essay question, long and short essay question. So study accordingly. In case of any doubt, contact me directly. Thank you.